Complex multicellular organisms contain several cells that must communicate with each other. This process of cell communication starts with one cell. We will call him Steve. Steve wants to communicate with this next cell. We will call her Mary. They want to communicate, but they have no mouths. So Steve lets off signaling chemicals called ligands to talk to Mary. How do those ligands get to Mary, though? Mary uses signal transduction pathways to get that ligand through the plasma membrane. The first way is through G-protein receptors. The ligand comes to a G-protein receptor and slides right into the space, like a car into a parking spot. An inactive G-protein carrying GDP comes along and the receptor activates it and causes the GDP to turn to GTP, like an on-off switch. An inactivated enzyme known as adenyl cyclase comes along. The G-protein goes to it to give it a phosphate from its GTP to activate it, causing the G-protein to become inactive again with GDP and the enzyme to change shape. This enzyme causes a cellular response to occur, aka Mary gets to hear what Steve wanted to say. There are two common second messengers that can carry out the response, cyclet AMP and the calcium ions. We can call them CAMP and the milk delivery guys. In this case, the enzyme will create CAMP. CAMP will now go and activate protein kinases. They transfer phosphates from ATP to protein. Then boom, the cellular response is accomplished. Mary knows what Steve said and now she can complete her task and be happy.
But let's say the G protein activates the phospholipase C, or PC, instead. In this case, PC will release IP3 to open the gated channel, releasing calcium ions. These calcium ions race out into the cytoplasm to find and activate proteins. These proteins then prompt a cellular response. Signal proteins attach to the active sites of each tyrosine kinase monomer, causing them to form a dimer. This signals ATP to attach its phosphates to each tyrosine, leaving ADP behind. A relay protein then binds to an attached ATP and becomes activated. Each activated protein triggers a transduction pathway, leading to a cellular response. The virus signal CD4 attaches to the cell's chemokine coreceptor and is consumed by the cell. It then releases viral enzymes and viral RNA. Viral RNA is reverse transcribed into viral DNA, which penetrates the nuclear envelope and corrupts the nucleus.
In the nucleus, the viral DNA is carried in by a viral enzyme. The enzyme attaches to a host chromosome to bring in that viral DNA. This causes HIV to spread rapidly through mitosis and affect the cell's function. The symptoms of HIV are fever, vomit, diarrhea, headache, and nausea. There is currently no treatment.